Good afternoon and welcome to today's webinar with Qlucore. My name is Mats Svensson and I'm going to be your host today. Normally I work here at Qlucore with development and support, but I also give some of these webinars, so you might have encountered me here before. Some of the names here I recognize from the support, so it's nice to see you guys. We got people from both Europe and the US at the moment. And today's topic for this brief webinar, we've changed to a briefer webinar format. Instead of 45 minutes, it's 30 minutes. It's easier to find a spot to listen to it. So the topic of today's webinar is dimensionality, and the title of the webinar is "What is the dimensionality of a data set?" And this is a rather mathematical subject. However, what I'm going to do today is try to give you a more pragmatic approach to this, a more practical approach to it, to tell you what you can use this stuff for. So, first of all, when we look at data, in Omics Explorer or in any other form of graphical representation, it's usually the fact that what we see is a simplification of the actual world, because the actual world is a lot messier than what we see in the graph. And what I mean with this is that what we see on the screen is a simplification. The PCA plot, for example, or as mentioned, any other graphical representation on a screen of most data sets is only showing you parts of the actual complexity of your data. And what do I mean with that? Well, what I mean with that is that the data usually has more dimensions than what you see on the screen. Typically, in our PCA plot, we show a three-dimensional plot, and the three-dimensional representation you see on the screen is not the exact image of your data, because your data usually has more dimensions than three. And if we divide this into what we say, when we say this mathematically, dimension means the number of orthogonal components defining the data space, or the number of degrees of freedom in a data set. So if your sample is a point in this n-dimensional room, the number of degrees of freedom is the number of coordinates needed to describe where that point is. And depending on your variables, whether or not they covariate, which they usually do to some degree, your variables, the number of variables is the number of dimensions. But that's not really necessarily the case, because the variables may be connected to some degree. Practically speaking, dimension means how complex is my data set? How much information is there to be found within the data set? And typically when you work with a PCA data set, the question you're looking for is how many sample clusters or how many groups of samples that are similar in some way exist in this data set. How many can I expect to find? Have I found all the clusters of information in my data, or have I missed something out because I didn't filter it well enough, or didn't take into account the complexity of the data set? Those are questions you can ask yourself. That has to do with dimension. So a simple data set with only three dimensions can be exactly represented in the PCA plot, whereas a higher dimensional data set can't. So the mathematical view on this is the PCA plot is again a three-dimensional projection of a higher dimensional data set. So we take something in higher dimension and reduce the number of dimensions to three. And since this is a projection, it only shows parts of the truth. You will lose information in that projection. The practical view to this, however, is that a PCA rendering onto three dimensions of something that is of a higher dimension often is enough to show whether there is similarity between two samples, and can therefore be used to detect clusters that are formed in higher dimensions. But there are a number of tricks we can apply to make this easier to us. And the first trick is the PCA plot itself, and I'll explain it using an analogy I usually use for this, and that's the shadow of a pen analogy. we view a shadow of an object. When light is cast and there is a shadow of an object, it's usually easier to see what that object is depending on the direction of the light. If you take this image, for example, on the left-hand picture, it's very clear that the person is holding a pen, whereas it's not obvious in the right picture that it's the same pen being held in front of the camera. So the direction of the light, the direction of the projection, determines how much of the information is preserved in that projected image. And that is exactly the same thing that happens in PCA, because if we choose the projection carefully, we will retain so much information that we can make it useful anyway, even though we lose information. And practically speaking, in Omics Explorer, this is done in the PCA plot, because the PCA plot always tries to use the best projection possible. However, there might be other projections that might be interesting, and also the PCA plot has a lot of other tools connected to it that can be used to find information about the dimensionality of your data set. I'm going to show you today in the demo part of the webinar a couple of plots called Talus and Scree, which are used to find out how many dimensions there are in a data set. And also, we'll take a look at something called the neighbor graph, which is a way to know a little bit more about whether your samples are similar or not, and to get more information than the PCA usually can convey. We'll also take a look at something called projection score, which is a method developed here at Qlucore that we use to find the best possible projection for a data set. So those are the methods available in Omics Explorer. Let's see here. There. Sorry, I was a little bit lost here. Another thing to ask oneself is the following: how many sample clusters can I expect from a data set? That's an important piece of information, because usually when we look at a data set and analyze it with exploratory methods, what we do is that we peel off one cluster at a time by filtering the data set in some manner. So what we can do, for example, is to do variance filtering and find the strongest signal relative to the other data. That's our first cluster. Then we remove that and repeat the process. That's a sort of simplified way of doing it, because usually if we want to do this in a stringent way that is scientifically correct, we should do a bit of statistical testing and so on as well. However, as I find these sample clusters, one good question to ask oneself is, how many of these clusters can I expect? And sometimes that question can be answered quite easily by the issue at hand. For example, if I am investigating samples containing a to me known disease like leukemia, and I know what subtypes of leukemia are supposed to be present in the data set, or which subtypes of leukemia are usually present, then I might have an idea. However, if I'm looking at a much more complex data set and de novo sequencing of stuff that I don't know anything about, then it's a lot harder. And then the Talus and Scree plots can help me. And I'll show that later on here. Also, another good question to ask oneself is how projection score shall be used for best results, because projection score is a tool that can help you find the so to speak optimal point where to stop doing variance filtering. Because when you're doing variance filtering, what you're doing is you're throwing away noise to try to find a signal. And when you're throwing away noise, you want to stop at some point, because at some point you're throwing away too much, and then you get a worse result than you would otherwise. And the whole trick here is to find a good cutoff point where to stop doing that variance filtering. And projection score is a good way of doing that. And dimension analysis can actually help you in some ways to improve the projection score projection score optimization. Another very good question is, and that's the real million-dollar question here, can I trust what I see in the PCA plot? That's a tricky question because actually, as I said, when you do a projection from a higher dimension to a lower one, well, what happens then is that your image is sort of distorted. You don't get the whole truth. And that means that artifacts of the actual algorithm can show, in the sense that you might see clusters that don't exist for real, false positives. Can I trust what I see in the PCA plot? I appear to see 6 groups, but the dimensionality analysis tells me that there should be no more than 4. Maybe I should be a little bit careful then, because then I might not be able to trust the PCA perfectly, and I might have to take another look at my data set. And we're going to look at the finer details of the PCA plot today as well, and see how the variance percentage that is presented by Omics Explorer can be used, and how the axis percentage can be used. And I mentioned this one before as well, the neighbor graph. The neighbor graph is a really, really useful tool. Okay, those are the scientific questions and the introduction to what I'm going to show today. And what we are going to do now is to move on to run Omics Explorer and take a look at some of these features that we can use. So let's go to Omics Explorer. This is Omics Explorer version 3.5, the latest version that is out there. And it's a good idea. I know that several of you are current users and not new users of Omics Explorer, so some of you are already using this software. There is going to be a service release of 3.5 very soon, probably next week. And that service release will correct a few issues that we've found in the software. So next week there's going to be a newsletter about that. Take your time and download the new version, because that's going to help you with some annoying little bugs that we have. Let's open a data set, and we'll open the standard leukemia data set that's shipped with Omics Explorer. Here it is. Here's the sample PCA view. You've seen that before. Let's launch the statistics window here and take a look at, oops, the statistics window ended up on my other screen. There we go. Let's take a look at a few features here of the PCA plot itself that has to do with dimensionality. Because, as I said, what we're doing here is that we're taking a higher dimensional problem and reducing it in dimension to the 3 dimensions we have on the screen. And mathematically speaking, we have 22,000 variables here. So we sort of have a 22,000 dimensional problem. However, the dimensionality of the matrix here is actually limited by either the number of samples or the number of variables. So in this case, we have 132 samples. So we have 132 dimensions that are being projected down to 3. So how can we get something 123 dimensional and show a good picture of it like this? Well, actually, what PCA does is that it takes the approach I did with the pen in the picture. It takes the best direction. So it's chosen a direction which maximizes the amount of information being kept. And information in this case is variance, because variance describes how similar or different the samples are. And we've chosen these 3 directions, which are orthogonal. This one is labeled 1, 2, and 3. And 1 has a number here, 21%, 2 has 7%, and 3 has 5%. There's no surprise in the fact that these are ordered. So this is bigger than this, which is bigger than this, because these 3 are our first 3 principal components. And the principal components determine the 3 directions in 132 dimensional space, which retain the most information. This one contains 21% of the total variance. This one retains 7%, and this one has 5%. And the sum of 21, 7, and 5 is 33. or well, it's 32 really. No, it's 33. Yeah, I'm correct. Sorry, it's 33, and here it says 33%. And 33%, this is the amount of variance from 132 dimensional space that we can show in this space here. So we've lost about 2/3 of the information. Is that useless to us? No, it's not, because if we start doing filtering by variance here, sooner or later what we'll end up with is two very clear main groups here, and one of them happens to be one of the main types of leukemia, and the other one happens to be the other category of lymphoblastic leukemia, which consists of several subgroups. So that's how these numbers on the PCA axis work, and the number down here, those are actually a reflection of how much of the information is being kept. And while we're at it, when we're talking about dimensions and PCA plots, it's worth mentioning that the PCA plot has a few options here. I can actually choose here which of the dimensions are shown on the axis. So here I can choose dimension 15, for example. Dimension 15 doesn't contain very much information, which is not very surprising, because these are sorted in order of relevance. So the three first ones usually contain the most information. But that means that you can choose another projection direction if you want to. And there might be reasons to do that. So that's the PCA plot. The next thing related to the PCA plot I want to show here is the neighbor graph, because the neighbor graph will give you even more information. Let's say that I've found these two main clusters, this big one and this small one, and I want to figure out, okay, these two samples are close to each other, so they appear to be similar. And these form a cluster here, so they appear to be similar. But how similar are they? Then I can use this network spinner here. It's on the method tab. The network spinner, if I change that to 1, what happens is that you see that there are connections in the graph. And the connection now is that each sample is connected to the one other sample which is most similar to it. And most similar means most similar in 132 dimensions, not in 3 dimensions. So what you actually get here is a graph that gives you more information than the PCA plot itself. If I choose the 2 most similar samples, you see there are more connections being made here. I can go to 3 and to 4 and to 5 and to 6. And when I go to 6, I get a connection from this group to this group. But if I choose the 5 most similar samples in 132 dimensions, the groups still seem consistent. So this gives me a lot of information that, okay, these samples really are probably significantly similar. It's worth going on with statistical tests and maybe other experiments and so on these, because there is really similarity between them. And as you see, there are very few connections between, let's see here, this subcluster here and the other subcluster. So you can sort of see on the graph here that the big group forms subclusters here, and that gives me a strong indication that there might be more groups in it. So that's the next tool you can use. Another really useful tool to figure out more about dimensionality of a data set is the Talus and Scree plots. And the Talus and Scree plots, they're available here under the plot subgroup here. You can either select the all here and select them from the palette of plots here, or you can select special and select Talus or Scree. Let's start with the Scree plot, because that's something well known in literature. It always looks something like this. It might vary a little bit, but it always looks like a slope, downwards slope. It always slopes downwards in some way. And the reason it does that is that this shows your principal components. Here is principal component 1, here's number 2, here's number 3, and so on. This shows you the percentage, the amount of the total variance that is retained by that component. And the first component here has got a high number here. It's about 15, 16%. The second is at 10 or so. If I move this back to where we started, we get the same numbers as we had on the PCA plot when we started. So it's 21% on the first one, and 7 and 5. And what we see here, which is interesting, is that the first three principal components contain very much of the information, about 33%, and the rest of it is here. It's very little in the last few ones here that's shown on the screen. However, if this slopes, and they're always sorted, so this always goes down. If this slopes less, there is more information in the further components here. And more information in the further components means that the data set is more complex, which also means you might expect more groups in it. And well, that brings us to the next type of plot, which is a modified version of this one. This is the Scree plot. The Talus plot is related to the Scree plot. It doesn't show percentages, it shows differences instead. So what we show is the difference between a principal component and the next principal component. And it's a logarithmized difference as well. So it sort of amplifies that difference. And here's an interesting thing that we can see here. But what's going on here? Well, we see that the plot fluctuates quite a bit here, but after this one, after the 7th or 8th component, after about the 7th component, there is sort of fluctuation along a line which slopes very slowly downwards if we would extrapolate this. Now, the last little peak we have is here on principal component number 7. It's not really very clear from the plot, but you can sort of imagine it. And what you also should note is that the scale on this side here is constructed so that it's a linear scale, but it's a logarithmized value. So here we actually have quite a bit of cutoff, right? So the last peak we have here is about 7th principal component. So with 7 principal components, we should expect about 7 or 8 groups. There's not much more information in this data set than about 7 or 8 clusters. The rest of it will be artifacts of the algorithm or artifacts of the human eye trying to find patterns in something that doesn't have more patterns in it. And if we take a look at the cheat sheet here, which is this is an annotation for the data set containing the different subtypes of leukemia that are present here among the patients. Here we have 1, 2, 3, 4, 5, 6, 7 groups. These are what we expect to find, and this we actually can see from this last peak here. The last peak will give me the notion that okay, here is really the extent of information carried in this data set. The rest here to the right represents dimensions that really contain only noise. There's so little going on in these dimensions, it doesn't really distinguish the samples from each other. And that's the real point here, because what we're doing here is we're looking for clusters and samples that are really distinct, similar or distinctly dissimilar. And again, we go to the PCA plot. Let's remove the network here so it's easier to see what is going on. There we go. Here's where we started. Here's the PCA plot where we started. Now let's do again what we did from the beginning, where I did variance filtering to find those 2 main groups. Now I do variance filtering with the variance tab on the statistics window here. So the variance tab, statistics window. It's new in 3.5 that statistics and variance are on different tabs. I start doing variance filtering and somewhere that other group appears. If I keep doing variance filtering, the result will deteriorate, and around here, the result is useless to me. So what I'm actually seeing here is an interesting biological fact, because what I'm seeing here is 2 very distinct groups. And I've filtered away a lot of noise, and I filtered away the distinction between the 2 leukemia groups, which I could see earlier on, there you have the two leukemia groups. And if I keep filtering, that distinction will go away and I'll have two distinct groups with very little information retained. What I'm seeing here, if I look at the variables here, I'll have to see the gene symbol. You see here, XIST. That's a gene that's related to the X chromosomes activation, which means that these are the boys and the girls. These are males and females. That's the strongest signal in the data set. That's just a parenthesis. However, I do my variance filtering here, and somewhere I detect that those two main groups. Where's a good point to stop filtering? Where can I say that, okay, this is a good filtering, this will give me a good projection, I can probably trust this. Back to, again, dimensionality analysis. We've introduced a tool here called projection score, which is this little thing here, the little box here. And this little box here will give you a score, and it will give you a score ranked on red, green, and yellow, green being really good and red being bad. Should be taken with a pinch of salt, because that's really depends on your data set. However, as you see, when I'm doing the filtering here, it changes. So if I start from the beginning here, I start with a projection score of 0.41, which the program says, okay, this is green, which is sort of good. I keep going, and as I keep going, it goes down. And here somewhere it goes up again. It reaches a maximum of 0.43, and as I go on, it quickly deteriorates again. And the maximum appears to be around 0.43. And here's a cool little tool. You can press the optimize button. There you go. There you get the best projection score. And this will give you what is probably a good cutoff for your data set. This is just a rule of thumb. There might be better projections for your data set to see those differences, because again, remember, we're losing information in that projection. There's another cool thing, which is the last thing I'm going to mention here. You can do with projection score related to dimensions. Here, you can change the number of dimensions that are taken into account to calculate the projection score. The default is 3, because that's the dimensionality of the plot you're seeing. You could change it, so you could possibly, there we go, we change it to, let's change it to 6. And that will give a slightly different result. If I optimize it, it ends up there, which is, since I know that I have at least 6 or 7 dimensions, this will show, actually, if we look at it, this cluster here is a lot clearer than it is if we do it with the exact settings we got from 3 dimensions. So by knowing the number of dimensions of my data set, I can get a more optimized view in this one by pressing the optimize button here and selecting 6 or 7 dimensions instead of 3. 7 doesn't work too well, and there's a good reason for that, because if we have 7 distinct groups, it's a 6-dimensional data set. So that was what I was going to show today. These features are really cool. You don't have to understand a lot of the mathematics behind it. It's just good to know what dimension does to your data set, and it's good to have a bit of understanding of this, and keep that picture of the pencil or the pen or the shadow of the pen in mind when you think about this, because that's a really good analogy, and it's a really good analogy when you try to explain this to people who are new to it. But if I choose the light direction correctly, I'll get a good picture of the pen. If I choose it badly, I'll get just a point or just a line, which is difficult to interpret and difficult to see that it actually is the pen I'm looking at. So that was what I was going to talk about today. Any questions? Feel free to ask questions in the chat, because there's a lot of people connected. I can't take questions verbally, so please input the messages to the chat, and I'll read the question to everyone and try to answer it. Anyone? I'll leave the chat open for a few minutes. Now, another thing to remember here is yes, I got a question from someone here. I'm quite new to Qlucore, so there's something I want to have explained to me. Yes, please. You said 132 dimensions are not variables dimensions and not samples. Well, yes, sort of. Sort of, yes, you're correct. However, what we're doing here is that if I go back to my presentation here, let's see, if I go back to my presentation here, that's very correct. Variables are sort of dimensions, but what is the PCA plot? Didn't I have a picture of that here? No, I didn't. Oh, sorry. What is the PCA plot? Well, the PCA plot is a rendering of my data set. But what is it rendering? It's really a rendering which is based on an analysis of the variance. So what I do is I could take the covariance or correlation matrix. I could do it with either of them, and use that matrix. By looking at the correlation matrix, I can take the eigenvectors of that matrix. The eigenvectors of that matrix sort of determine the orthogonal components that create n-dimensional space for it. But the covariance and correlation matrices, they are square matrices, and those the dimension of that matrix will be the minimum of number of variables and number of samples. So yes, it's 132 dimensions, even though I have 22,000 variables. I hope that explained it to you. It sounds a little strange to start with, but imagine the following. Okay, I have several thousand variables, but those several thousand variables determine where those 132 different points end up. So even if I have a lot more variables than I have points, it's sort of the points that determine how big the problem as such is, in layman's terms, because I'll have a square matrix when I do this. Okay, the person who asked the question says it's clearer now. Anyone else got anything? Okay, I should just remind you that if you come up with any questions after the webinar, or if you try these things out and have questions on how to get them to work, the webinar as such will be available on our webpage in a few days. And you can always send an email to support@qlucore.com if you have any questions regarding these things and want to have help getting started with them, or if you get stuck on some of the things here. I know also for a fact that there are a couple of really good white papers on our webpage on this. I don't know exactly where they are, but they are in the white paper section. I know there's at least one which is very good at explaining this. And also, of course, the manual in the program has help for the Talus and Scree plots where you can find the exact mathematical definitions of them. And also there's a couple of references if you want to quote the Talus plot, for example, which is a new version of the Scree plot that we introduced. With that being said, I hope this was useful to you and I hope it was fun. A lot less biology than most of you do, perhaps, but it's a bit of mathematics today. So with that being said, I wish you a good day and I hope to see you on the next webinar. Thank you very much.